Genome evolution due to indels. When the ancestral sequence is not known, insertions and deletions cannot be distinguished from each other, then they are called as indels. So as if we do not know the ancestral genome, so we cannot distinguish that whether there is a insertion or there is a deletion. So such insertions or deletions, they are called as indels. In other words, when one cannot infer the phylogenetic direction of the sequence change, the sequence change event is called as indels. So when we cannot direct that how there is a change in the sequence of DNA, so such changes that can be the insertions and they can be the deletions are called as indels. A microindel is defined as an indel that results in a net change of 1 up to 50 nucleotides. If there is deletion or insertion from 1 nucleotide up to 50 nucleotide, then such indels are called as microindels. In the coding region of the genomes, unless the length of indel is a multiple of 3, it will produce a frame shift mutation. Let's say if there is indel of 1 nucleotide or if there is an indel of 2 nucleotide, they will produce frame shift mutation. But if there is indel of 3 nucleotide, then there will be no frame shift. Rate of indels in most genomes, including humans, tends to be lower as compared to the base substitution. So their rate in case of mammalian genomes or in case of eukaryotic genomes is less when we compare it with substitutions. Each person has approximately 192 to 280 frame shift indels. Indels form an important source of sequence change that is actually the source for genome evolution. Most small indels are in fact deletions in the form of 3 nucleotides. Indels can have any size, even up to several MBs in size. The majority are single nucleotide indels. This is the diagram to understand that what are indels. Indels, here we can see that in this sequence, 2 nucleotides, they have been deleted. Here we can see that 3 nucleotides, they have been deleted. Here we can see 4 nucleotides, they have been deleted. If there is deletion of the nucleotide or there is insertion of the nucleotide, and we cannot depict about that whether it is the insertion or deletion as compared to the ancestral genome, then they are called as indels. So here wild type sequence, we can see that this is the wild type sequence. Here there is a 3 base pair deletion or there is a indel. So here we can see that 3 nucleotides, that is T, A, this has been deleted from this sequence. So this is the indel. And same here we can see that 4 base pair has been inserted. So these are the 4 nucleotides that have been inserted in the original sequence, in the wild type sequence. So here indel can be in the form of deletion and indel can be in the form of insertions. Indels in case of human genome, in human genome there are approximately 7.1% indels, including all types of the variants. Recombination between direct repeats in a single chromosome leads to potentially large deletions. Indels can be in the form of single nucleotide and it can be in the form of large deletions. So large deletions is usually due to the repeated sequences which are present on the genomes. So recombination requires sequence identity over fairly large regions. So these deletions are mostly large because the sequence similarity is large, so the deletions, they are also large. Unequal recombination also lead to the insertions or deletions. Unequal recombination can lead to insertions, particularly which are also called as segmental duplications. This is the example of segmental duplication, where we can see that there is unequal recombination. So here we can see that this is unequal recombination between the sister chromatid. This is sister chromatid 1 and this is sister chromatid 2, and we can see that the crossover is unequal. Same process can also happen between the parental homologous chromosomes. Here we can see that how unequal recombination. This is the original DNA sequence where box has been marked as C, B, C and D, but as a result of unequal recombination, we can see that A, B, C, D, E and F. This is the original DNA sequence. Then this B, C, D, E and F is replicated from the second strand, is inserted from the second strand, and ultimately this is the G. So this is the duplication between the sister chromatids, where we can see due to unequal crossover, this fragment of the DNA now is present on the original DNA sequence. So unequal recombination is a phenomena by which indels can occur. Genome evolution indels. If we conclude that what are indels, when the ancestral sequence is not known, insertions and deletions, then they cannot be identified, they cannot be distinguished because we do not know the ancestral chromosome. Then such insertions and deletions are called as indels.